So our next speaker is Llewellyn, and she's from Northern Arizona University. She's going to talk to us about Pinyon Stomata.
Hi everyone, my name is Tara, and I'm actually a junior right now at NAU studying environmental science management. And my mentor, Dr. Llewellyn, and I did a project of heritability of Pinyon pine stomata. And why we chose Pinyon pine, it's a very important part of the local ecosystems in the Southwest, and especially in Northern Arizona. So here's just a brief overview of what I'm going to talk about. So a short introduction with some background information, our hypothesis, methods and materials, which is our cone collecting, and genetic testing, the imaging system I use to take the pictures, and the imaging program, which is the computer software I use to analyze to get my data, our results, and the conclusion. So our introduction, as many of you know, climate change is projected to limit already scarce water resources in especially the Southwest. And this picture actually is a very common sight, unfortunately, especially at our sample sites, which is located in Sunset Crater. Adult pinyon pine trees are no longer surviving at elevations that they once used to due to the drier climates. They just aren't adapted for it. So we're really interested in looking at the ones that are surviving and what traits they're passing along to their offspring that make them more drought tolerant in the drier climates. So I just kind of put up a definition of heritability, which is a variation in a particular trait that can be attributed to inherited genetic factors. And the point I wanted to make with this is the most common example you hear is a person's eye color. But stomata traits are more like a person's height, where although genetic factors play a really important role, so do the environmental ones. So one way that pinyon pine trees may be adjusting to the climate is by altering their stomata traits. And this is kind of a picture. Imagine that's a leaf. They actually have needles, not leaves. And this is actually not really drawn to scale because this only shows that there's 3 stomatas. And stomata, or stomates I should say, stomata are microscopic pores that exist on all leaves and needles. And it's pretty much where they absorb carbon dioxide and release H2O to the atmosphere. And I know this shows only 3 of them, but in reality, every leaf or needle has up to hundreds. So there's quite a bit of these little microscopic pores. And there's a balance between the stomata because when they open, they do absorb more carbon dioxide, but they also release water. So they have to have a really good stable balance between the two. And so our hypothesis for the project is that there will be a correlation between the offsprings and the parent stomata measurements. And a significant relationship between the trait of the parents and the offspring suggests the trait is inheritable. And the one we specifically looked at was the surface area of each individual stomata. And if the trait is inheritable, it can evolve in response to climate change to increase water use efficiency under climate change. So our methods and materials. So cones were collected to determine the mom, and then genetic testing was to determine the dad. The mom was really easy to find. You kind of just pick up the cones, and they're always up in the moms. The dad was actually a really exciting project that one of the grad students in the Pinyon Pine Research Group did. And so what she did is she collected those cones, took the seeds out, and then they planted the seeds in our greenhouse garden, and so they grew them for 2 years, and then we ended up transplanting them into Sunset Crater where the parents are. And what she did, she took needle samples from all the offspring and the parents and did genetic testing on them. And we found some interesting things. First of all is some of the trees actually selfed, which means the mom is the dad, that's the mom, which doesn't usually happen. And the other really interesting thing they found was there were brother trees out there, so we weren't 100% sure which brother it was. So we made sure when we picked our samples that we knew we were 100% sure who the mom was and 99.9% sure who the dad was, so we for sure knew them. And after the parents were identified, samples of the needles were collected to be used for, it's called the Visionary Digital Imaging System, and this is kind of my part of the project. And there's a picture of the equipment, and it really does have like a dozen components like that. So what it really does is it allows me to get really close, so microscopic level, with really high resolution cameras to take pictures. And that's what I did for each sample that I had for each individual. So here I'm going to show you 2 examples, and I apologize if you can't see all those little holes, little circles, but those are stomata, and as you can see there's 2 needles. Pinyon pine, or Pinus edulis, is known for having 2 needles per fascicle. So what I would do with a fascicle is I would take it and I split it up, so I tear them apart. One would be face up, and one would be face down. The face up one is actually the one I use during analysis because they're more open, so you can actually distinguish the circles. You can also see from this example that this is one of the offspring, so it has 2 complete lines of stomata and then 2 incomplete, so that's just an example of one of them. And then in example 2, this is a different offspring with different parents. They actually have 6 almost complete lines. So not only did the lines change, but the amount of wax, which is the white stuff you see, can change, along with even the color and texture of the needle depending on the parents. So ImageJ is the computer software I use to get my analysis of the surface area of the stomata. And it's a free Java-based imaging processing system, but pretty much what I did is I would upload one of the photos, I would crop it, and they would all be the same crop size, and then I'd have to pretty much invert it, make it binary, and a few other little steps, and then it would allow me to look for circles is what it was trying to detect. So I would set the circle parameter, and it would detect those, and then measure each one that it could detect in pixels. I then averaged that for each one, so all the offspring and the parents, and then made my data. So here's our results. And as you can see, there is definitely some correlation between the offspring and the parents. I haven't completed all the samples yet. The more samples I seem to put in, the stronger the correlation is. So we're really excited. We're still trying to complete that to make a whole data set. But there's definitely a correlation between the offspring and the parents' average surface area of the stomata. So in conclusion, we conclude that stomata traits, such as the surface area, are inheritable, and that these traits are expected to evolve from natural selection. And I made sure to include a happy one with surviving Pinyon. So, yeah, and then acknowledgements, I definitely have to thank the Arizona Space Grants Consortium. Nadine Barlow and Kathleen Stigman, definitely, you guys were such a great help. My Dr. Amy Whipple, who unfortunately couldn't be here today, but she's really great to work with. The Pinyon Research Group for sharing their data, and Neil Cobb in the biology department for allowing me to use their new imaging system. So, are there any questions?
Since you're comparing parent plants and their offsprings, they're different ages, does that make any difference in the average surface area?
It would if it was the offspring, but the offspring all are roughly the same age because they were planted at the same time. But what we've seen with the parents is, even though they're older, depending on who, what dad it is, what mom, it mostly changes when they're growing. So the stomatas usually, that's when the average density and whatnot, that tends to evolve as they're growing, depending on how old they are. But once they reach maturity, it's pretty much the same. So.
Is there a possibility that environmental factors could influence the size of the stomata? Because if you're growing them in a greenhouse, that's different.
Yeah, absolutely. So what we tried to do is, the parents, they were already out there when they started this project. But the offspring, we tried to make same treatment for all. So they were all grown in the greenhouse for a certain amount of time, all got the same water treatment. And then we moved to the Sunset Crater Common Garden. It's in a very small contained area, and so there's very little where like this got more shade than this one. So we try to even that out as much as possible. But of course, there'll be some environmental factors, absolutely. So, yeah.
In addition to the surface area of the stomata, do you also look at, like you were showing the row things, and you're saying how some are complete and some are incomplete? Do you also look at that?
We actually, yeah, we've decided this summer we're actually going to expand on the project. So it's one thing we're looking for. The other thing is we want to get more measurements, so the length and width. The Java-based program, ImageJ, it was great to begin with, but we found some flaws along the way. So we're trying to actually find a better program that will give us more measurements on like the average length and width and stuff like incomplete lines. And so we're looking at, and we're also looking at the observations because we thought it was really interesting that based on the parents, even the texture and color changed. So we're kind of really interested to see why that is. So.
Is there a specific reason why some of them have incomplete lines?
You know, I actually asked my mentor, and there's really no reason for it necessarily. It just kind of happens. So we're looking into it. And the really funny thing too is when I was getting my samples, my fascicles, some of the trees even had 3 needles per facet, which they're not supposed to have. So that was even a potential new project, is looking at why some have 3 and not 2. So yeah, nature's nature, I guess.